Standard 8, Subject Science. Boys, today I'm going to teach you Chapter 10, Cell and Cell Organelles. Now over here, the question is asked in your textbook on page number 67. It is said, how many types of cells are found in the living organism? So, cells are of two types. You can see the pics on the left hand side of the slide. And those two types of cells are the prokaryotic cells and the eukaryotic cells. Now, over here, if you go to see, in the prokaryotic cells, the plasma membrane, cytoplasm and nucleoid, they are the only components. Whereas in the eukaryotic cells, all the organelles, they have a membrane. So it is a membrane bound organelles which are present in the eukaryotic cell. For example, I'll give you the organelle like in case of nucleus in the eukaryotic cells. This nucleus has a covering that is called nuclear membrane, and inside the nuclear membrane you can see the dark colored structure, that is nucleolus, that is there present. So it has a membrane, whereas in the prokaryotic cell the nucleus do not have a membrane. So there is, in short, you can say they do not have a well-defined nucleus in the prokaryotic cells, whereas in the eukaryotic cells they have a well-defined nucleus. This you can remember. Then it is asked also, which instruments are used to observe the cell. On the right hand side there is a picture of that. It is a microscope. So microscopes are used to observe the cells which are minute in size, and those are micrometer and nanometer. They are the units used for measuring their sizes. So microscope is used to observe that is the compound microscope. Compound microscopes are used to observe the cells. If you want to study anything in detail, then you study with the help of the compound microscope. And if the cells are so small that you cannot see it with your naked eye, neither with the compound microscope, then we use electron microscope. Structure of the cell. You can see the plant cell and the animal cell. If you see the plant cell, it has the cell wall. But in animal cell, the cell wall is not seen. It is absent. Now this cell wall over here, it gives a definite shape to the cell. Now you will see on page number 67 of your textbook, there are three columns given. The first one is the component. Second column is the animal cell. Third is the plant cell. So over there, you have to write down. So you can take a pen or a pencil with you and you can write it down by looking at the diagram shown on the slide. The first one, if you go to see the cell cell membrane, if you see in the plant cell on the right hand side, just below from the, that is number 4, you can see the cell membrane shown. Whereas in animal cell also, cell membrane is shown on the right hand side of the diagram. So the answer is present in plant animal cell and present in plant cell. Now the next one is a cell wall. Now this cell wall, it is, if you go to see, it is absent. You will not see the cell wall in animal cell. So the answer is absent. Where in plant cell, you can see the cell wall shown on the right hand side. Number 5 from the bottom, you count. So you can see. So cell wall absent in animal cell, present in plant cell. Then there is the next one. The third one is lysosomes. Now these lysosome, see in the animal cell, the spelling is there, lysosome. I'll be explaining you in detail about all these organelles. Now you only write down the, just you have to fill up that. So lysosome, they are present in the animal cell. But you will not see lysosome in the plant cell. If you see it clear on that plant cell diagram, it is not there. So it is absent. Then the next one, the number 4 is a plastids. Plastids are like, chloroplast. So chloroplast is not present in animal cell. So it is absent. But it is present in the plant cell. You can see the chloroplast written over there on the right hand side from the starting number 4. That is rough endoplasmic reticulum is there, then Golgi, then smooth endoplasmic, and then after that comes a chloroplast. So chloroplast is present only in the plant cell and absent in animal cell. Then after plastids, you have to write down endoplasmic reticulum. Now endoplasmic reticulum, see if you go to see in animal cell, it is present on the right hand side of the diagram. Whereas in the plants also it is present. And they are, in plants they are of two types. One is a rough endoplasmic reticulum and the second one is a smooth endoplasmic reticulum. That I will explain you when I am teaching you about each organelle. Now only remember that it is present in both animal cell as well as in the plant cell. Then the vacuole. If you see the vacuole over there, now these vacuoles, they are present in both the cells. That is an empty cell which is present in both the cells. That is, you have to write down present in plant cell, in animal cell as well as in the plant cell. So they are present in both. That is the vacuole. Then comes the next one, Golgi complex. Now this Golgi complex, or it is called as a Golgi apparatus. It's the same. Golgi complex or Golgi bodies, Golgi apparatus is the same. So you will see that it is present in plant cell as well as in animal cell. Then comes the mitochondria. Now if you go to see mitochondria, singular it is mitochondrion, but when it is two or more, then it is called mitochondria. So if you go to see in both the diagrams, mitochondrion is present. So now you will be wondering like the saying, vacuole is not seen in animal cell. It is not marked over there, but it is very small in animal cell, which is not shown. Actually it is there in animal cell, don't worry. And in plant cell it occupies a big space, that is a vacuole. You can see in the plant cell that central vacuole from the bottom, the third number. The vacuole is very big in plant cell, but it is very small in animal cell. So I hope you have understood that chart which is given. Now you have to write down the fill up. There is a plant cell and animal cell diagrams are there, that you have to write it. That I'll be sending you the names. You can write it down also as I'm explaining. On the left hand side, the first one is a mitochondrion. After that comes the cell wall. Then it is a cell membrane. Then comes the nucleolus, that is a purple color. Green color, that is actually it is not, that is they have shown the color, that is cytoplasm. And the next one is the Golgi bodies or Golgi complex, whatever you can write it. On the plant cell on the right hand side, you can write it as endoplasmic reticulum. Then comes it is a chloroplast, and after that is the vacuoles. So you can see over there the pic on the slide, and in the textbook also you can mark it over there, both for the plant cell as well as for the animal cell. Component of a cell. So I'll start with the cell wall. First one. Now this cell wall, if you see in the diagram, it is marked over there from the bottom, number 5. Now this cell wall, it is seen in the cells of algae, fungi and plants. Now the algae, for example, are Spirogyra. Fungi, they are the mucor. So in that, and plants. So the cell wall is found in the cells of algae, fungi and plants. But animals, they do not have cell wall. So animals lack the cell wall. So instead of cell wall, then what do they have? They have the plasma membrane or the cell membrane, which I'll explain you after that. So animals do not have cell wall. And how does a cell wall looks in the plant cell? It is elastic, strong, and it is made up of carbohydrates like cellulose. So cellulose is a carbohydrate. Now you must be wondering, what is carbohydrate? Carbohydrates are the one which is made up of carbon, hydrogen and oxygen. So the cell wall, it is elastic, strong, made up of carbohydrate like cellulose and pectin. Now what is the function of the cell wall? Function of the cell wall, it is to protect and supports the cell. Now protect. Now they protect the cell by preventing entry of more water into the cell. So they control the amount of water. So they do not allow too much of water to enter into the cell. So that is, it is said that cell wall protects the cell by preventing entry of excess water in the cell. The first component of a cell was the cell wall. Now the second one is the plasma membrane, or it is also called as the cell membrane. Now this plasma membrane, it is very thin, fragile, very delicate and elastic. And this plasma membrane is the outermost covering for the animal cell. Now this plasma membrane is also said to be selectively permeable membrane. Why it is called? Because it allows some substances to enter the cell, while it stops or prevents the other substances. Now you can see over here, due to useful molecules of water, salt and oxygen, they enter the cell. And what is given out from the cell is the carbon dioxide. Now this plasma membrane, it also maintains homeostasis. Now what that means, homeostasis? That if any changes takes place outside the cell, the cellular environment does not change. Why? It is because of the plasma membrane. So this condition is called as the homeostasis. So plasma membrane, if you see over here, it is said to be a selectively permeable membrane, and also it maintains homeostasis. Then there are some activities that use the cellular energy, and those are, the first one is the endocytosis, and the second one is the exocytosis. The word endo meaning over here, it is the entry. It is the entry of some substances into the cell. And from where that comes? From the outer environment. So that means it gulps the food and the other substances. So it is the entry. Whereas exocytosis, exo meaning removal. That means it excretes the waste material out of the cells. So for these activities, the cell requires the cellular energy. Now there are the processes that don't consume the cellular energy, and that is diffusion and osmosis. And also in your book it has been asked on page number 68, how do substances travel in the cell? So they travel in the cell by diffusion, and second is osmosis. Now what is diffusion? Diffusion is like oxygen enters into the cell, and what is given out? Carbon dioxide. So it is an entry or exit of small molecules like oxygen and carbon dioxide. Now osmosis, it is the traveling of water or the movement of water from a part with more water to a part with less water through a selectively permeable membrane is called osmosis. For example, if I tell you raisins, dried raisins. Now when you soak it into the water, what happens to the raisins? It swells up. So that way there is, so osmosis, it is a physical process with three possibilities that I'll explain you. Osmosis, osmosis, it's a traveling or the movement of water from a part with more water to a part with less water through a selectively permeable membrane is called osmosis. Now you will see there are the diagrams shown. The first three, upward one, that is the osmosis in plant cells, and below that is the osmosis in animal cell. Now this osmosis takes place, it's a physical process with three possibilities. The first one over there, now if you take in your book, in your book it is shown isotonic, first isotonic solution. Now isotonic, and you see in the pic on the slide, the middle one, isotonic of the first one is the plant cell and the second one is the animal cell. So you will see that the medium outside the cell and inside the cell, they have the same proportion of water. Water does not go in or out. You can see that in both. Second is the hypotonic solution. There it has been shown on the slide. In hypotonic, what happens is the cell has less water. When the cell has less water, you can see the two arrows. In the first one, first part, number 3, it is written over the hypertonic. You can see the yellow arrows. That means the water is entering into the cell because the cell has less water than the outside medium. So water enters the cell. You can see it in the plant cell also as well as in the animal cell down in the piccoli on the slide. So that arrow which shows inward, meaning water enters the cell. So that is called as the hypotonic solution. Like I told you, for example, if you keep the raisins in the water, after some time what happens? They swell and they become turgid, full of water. The next one, the third one is so that process is called as endomeiosis. Why? Because the cell has less water than the outside medium and so the water enters the cell. Since the water enters the cell, it is called as endomeiosis. Third one is the hypertonic solution. Look over there, the arrows are coming out in the plant cell also as well as in the animal cell. Why? Because the cell has more water than the outside medium. So that's why what happened, the water come out of the cell. So this plant cell or the animal cell, if they are kept in the hypertonic solution, the water comes out of the cell. So that process is called as exomosis. And so there is a contraction of the cytoplasm. If you see in the plant cell also as well as in the animal cell, the cells they shrink because the water comes out of the cell. So you have seen over there, first one is the isotonic solution where the medium is the same outside and the inside the cell. The water does not go in or out. Then the second one is the hypertonic solution where the cell has less water than the outside cell. So the water enters the cell. So it is called as the endomeiosis. And the third one is the hypertonic solution where the cell has more water. So naturally it will come out of the cell. So that process is called as the exosmosis. The third component of the cell is the cytoplasm. Now cytoplasm, it is a fluid present between the plasma membrane and the nucleus. And many organelles, cell organelles like mitochondria, chloroplast, vacuoles, they are all being suspended. They are present in the cytoplasm. So cytoplasm is the medium for cellular chemical reaction. Now, the part of the cytoplasm other than the organelle, that is called the cytosol. So if you go to see, the cell is completely filled with the cytoplasm and inside the cytoplasm there are the cell organelles. So the part of the cytoplasm other than the organelle is the cytosol. So what does the cytosol does? That means cytosol is present in the cytoplasm itself only. So it stores the amino acids, glucose, vitamins. So this is a function of the cytoplasm. So in the cytoplasm, cytosol stores amino acids, glucose, vitamins, etc. In the animal cell, cytoplasm is granular. But in the plant cell, it is cytoplasm, it is very thin and it is pushed to the periphery. Periphery meaning at the edge of the cell. See, if you see in the cell structure of the onion peel on the slide only, the cell structure, the bottom one, you will see the cytoplasm. It is marked over there. Where it is? It is at the periphery, meaning at the edge of the cell. Why? Because the vacuole only is occupying the central part of the cell. So as a result, the cytoplasm gets shifted. It is get pushed towards the periphery. So that is about the cytoplasm. Already I have explained you about the cytoplasm. Now inside the cytoplasm, cell organelles are present. So the first cell organelle is the nucleus. You can see the pic over there. Now this nucleus, when it is seen under the electron microscope, it has two layers. You can see there, it is marked over the outer membrane and the inner membrane. So it is a double layered membrane. And those pores are there, the openings are there. So these pores, they allow the material to move in and out of the nucleus. Now this nucleus also has one round nucleolus. You can see it is marked over there in the diagram on the slide. So it has one round nucleolus and a network of chromatin fibers. Now what is the function of this chromatin fibers? What they do is, at the time of the cell division, they come together to form thick chromosomes. And these chromosomes are present in the nucleus itself only. Now, the functional segments on the chromosomes are called genes. So, chromosomes are nothing but they are the thread-like structures which are present in the nucleus. And on that chromosome, there is a segment, functional segment which is present. And that is called as a genes. And what they do, they transfer the hereditary characters from the parents to the offspring. So over here, for example, if your parent has blue eyes, the kids also will have blue eyes. It's just an example which I have told you about it. So, the functions of the nucleus is it controls all the metabolic activities of the cell and also the cell division. It transmits hereditary characters from parents to offspring. So this is about the first cell organelle, that is the nucleus, which I told you it is a double layered membrane with small pores, and those are called as a nucleopore. Why nucleopore? Because it is seen in the nucleus. So it is a nucleopore, and these pores, they allow the passage of material in and out of the nucleus. Also, the nucleus has one round nucleolus and the chromatin fibers. So this is all about the nucleus. The second organelle is the endoplasmic reticulum. Now this endoplasmic reticulum, it is an organelle which supplies or which carries the various substances inside the cell, is called endoplasmic reticulum. And it is a net-like structure if you see over there in the pic. And it has been filled with a fluid. Now this endoplasmic reticulum, it is connected to the nucleus always you will see endoplasmic reticulum is always connected to the nucleus from inner side, whereas the outer side is connected to the plasma membrane. Now this endoplasmic reticulum, the function is it supports the cell. It supplies the protein. That means there's a conduction of proteins. And if any toxins enters the body through food, air and water, it has been flushed out of the body with the help of the endoplasmic reticulum. So the first organelle what we studied is the nucleus. Second organelle is the endoplasmic reticulum. Both of these organelles are present in the cytoplasm. The third organelle, cell organelle, is the Golgi complex. Camillo Golgi, he described the Golgi complex for the first time. And this Golgi complex, it is made up of, you can see in the diagram, 5 to 8 hollow and flat sacs placed parallel to each other. Now these sacs, that is cisternae, it is called as, they are filled with different enzymes. So this cisternae, they work like a packing department that packs and distributes the substances. Now what is the function of the Golgi complex? This Golgi complex is the secretory organ of the cell. It also modifies, that means over here when I said it is called the secretory organ of the cell, meaning different secretions are prepared in the Golgi complex. So it is called secretory organ of the cell. Now these secretions, they are being modified and sorted out as per to their functions. And so they are further packed. So Golgi complex also produces vacuoles and secretory vesicles. Now what is secretory vesicles? They are the tiny little packages you can see in the diagram. They are the tiny little packages in which the cell secretions can be transported. So the first function of the Golgi complex, on the right hand side it is there, I have written, it is a secretory organ of the cell. Also it produces the vacuoles and secretory vesicles. Third, it helps in the formation of cell wall, plasma membrane and lysosomes. So these are the three important. The fourth one also is there. You can say that is the secretions are modified and sorted out as per their functions. And then they are further packed. Like for example, that is enzymes, mucus, proteins, pigments, etc. They are sorted out. And then they have been sent to various regions like plasma membrane, lysosome. So only you just remember there are four functions, at least you remember the three which I have written on this slide. It is the secretory organ of the cell, Second, it produces the vacuoles and secretory vesicles. And third, it helps in the formation of cell wall, plasma membrane and lysosomes. Fourth cell organelle is the lysosomes. Now this lysosomes, if you see in the diagram shown on the slide, they are the simple single membrane. They don't have double membrane. They have a single membrane bound sacs. And they are filled with the digestive enzymes. That is about the structure of the lysosomes. Now what is the function? The function is the first one. It acts as a immune system. That means it destroys the viruses and bacteria that attacks the cell. So that's why lysosome acts as a immune system. Second is it acts as a demolition squads, meaning it destroys the worn out cellular organelles and organic debris. So the process is called as autolysis, meaning self-destruction. So that is the second function of lysosomes. So the first one, which I told you, is the immune system, because it destroys the virus and bacteria that attacks the cell. Second, it acts as a demolition squads because it destroys the worn out cellular organelles and organic debris. So that process is called as autolysis. The third function, lysosome acts as a suicide bags. Why? Because when the cell becomes old or it has been damaged, then this lysosome, they burst and enzymes digest their own cells. You can see in the diagram, inside the lysosome there are enzymes which digest their own cells. Number 4, the fourth function of the lysosome is, that is, during starvation, lysosomes, they digest the stored proteins and fats. So that is the fourth organelle, that is lysosome. The first organelles what we studied is about the nucleus. Second, endoplasmic reticulum. And I told you it is always connected to the nucleus. So that's why first is the nucleus, third is the endoplasmic reticulum, and third is the Golgi complex. This discovered by Camillo Golgi. And he also won the Nobel Prize also. And the fourth organelle is the lysosomes. Number 5, mitochondria. It's a plural form. Whereas if you say single, then it is a mitochondrion. Now on the right hand side, there is a diagram shown on the slide. Now this mitochondrion, I'll say, because I've shown the diagram, it has a double membrane structure seen under the electron microscope. So you can see on the slide the outer membrane as well as the inner membrane. Now the inner membrane, it is deeply folded. That blue color is the inner membrane which is folded. And these folds are called cristae. And they are filled with proteinaceous gel-like matrix containing ribosomes, phosphate granules and DNA. That means it can produce its own proteins. Now, with the help of these enzymes, mitochondria, they oxidize the carbohydrates and fats. So when they oxidize the carbohydrates and fats, energy is given out. And that energy which is given out, it is being stored in the form of ATP. Now the full form of ATP, it is adenosine triphosphate. Now these plant cells, they have less mitochondria and more in the animal cells. Why is it so? Because if you go to see, the animals, they keep on moving and they need more energy for walking, running, all the other activities. So they require more energy. So they have more mitochondria. Whereas the plants, they are stationary, meaning they are fixed. They do not move. So they don't need energy to a larger extent. So therefore, plants have less of mitochondria. And this mitochondria, it is also called as the powerhouse of the cell because it oxidizes carbohydrates and fats. And so as a result, energy which is given out, it is in the form of ATP. Now what is the function of the mitochondria? First, it produces ATP. ATP meaning adenosine triphosphate. That is a full energy rich compound. Second function of mitochondria is it helps in the forming the proteins, carbohydrates, lipid, etc. by using the energy in ATP form. So this was the fifth organelle, that is the mitochondria. Now the next one, what I'm going to teach you is the vacuole. Sixth organelle, cell organelle, is a vacuoles. You can see on the diagram it has been shown the vacuole in the plant cell. It occupies the central portion of the cell. They are the storage sacs for solid and liquid contents. And it has only single membrane. Now these vacuoles, they do not have a typical size or shape. Why? Because the vacuoles, they change their shape and size as per to the need of the cell. So they do not have any fixed shape or size. Now the function of the vacuole is the first one. It maintains the osmotic pressure of the cell. Second, it stores the waste products and food. But in the case of amoeba, it stores the food before digestion. And in plant cell, vacuoles are full of cell sap. Sap is a white color sticky substance. So in plant cell, vacuoles are full of cell sap and it provides turgidity, meaning it is swollen up, and rigidity, it becomes strong. So in plant cell, vacuoles are full of cell sap and it provides turgidity and rigidity. That is about the cell organelle called vacuole. That is number 6. 6 is the vacuole. Then I'm going to explain you about is the plastid. Plastids. It's the last organelle. Now these plastids, they are of 3 types. Now what? Leucoplast, chromoplast and chloroplast. Now what is a plastid? It is an organelle but which gives colors only in the plant cells. Since they gave color only in the plant cells, so this organelle is called as the plastid. And this plastid, they have two membranes. You can see on the diagrams shown on the slide. Outer membrane and the inner membrane. And they are of 3 types. So the first one is a leucoplast. Now this leucoplast, or you can call it as a colorless plastids. That means they are white or colorless plastids. Now the function of this leucoplast is it synthesizes and stores food like starch, oils and proteins. The chromoplast, they are the colored plastids, because if you see on the chart shown on page 73 of your textbook, the first part, the first column is the color of the plant parts and the second is the pigment. So the leaves are green. Why the leaves are green? Because they have a pigment called chlorophyll. Same way carrots. Carrots are orange in color because they have the pigment called carotene. You must have seen the papaya plants, their color, papaya fruits, it is yellow. So that is because of the pigment xanthophyll. Then there is a color of the plant part that is purple or blue. Why? It is because now you have seen the brinjal, the colors are purplish because of the pigment called anthocyanin. Then there is dark pink, that beetroot shows that color. That because of the pigment called as the betalains. So these are the pigments, that is chlorophyll, carotene, xanthophyll, anthocyanin and betalains. Now you must have seen that ripe tomatoes, they appear red. But in the beginning, when green tomatoes, they become ripe, they lose their chlorophyll. And then they develop the red pigment in them. And that is because of the lycopene. Therefore, the ripe tomatoes, they appear red because of the pigment present in them, that is lycopene. Then the next one is about the chloroplast. Now this chloroplast, they are very important for the photosynthesis that takes place in the leaves. So what does a chlorophyll does in the chloroplast? It traps or it catch hold of the solar energy and change it into chemical energy. Then you can see also stroma, which is seen in the diagram. That stroma, they contain the enzymes, DNA, ribosomes and carbohydrates that are necessary for photosynthesis. Now, what is the function of the chloroplast? The first one is it changes or it converts the solar energy into chemical energy. During which process? During photosynthesis. So this you have to remember. And the last one is about the prokaryotic and the eukaryotic cell, which I have explained you at the beginning only of the topic. So now this topic gets over. I hope you have understood, boys, this topic. It's better you read the lesson and understand, and on the very same day itself I'll be sending you the notes in the assignment link. So kindly go through it and write these answers, question answers in your notebook. Thank you, boys.